Okay, so just to give you some context or where we are, it is surprise to no one that we are in a difficult planetary situation. So it's not only that we had COVID at some point that was really bad. This has been followed and probably will be followed later by a recession. We have major international problems and other threats. And then on top of that, we have this permanent threat of climate change. So many of these problems, in particular climate change, are to a large extent caused by emissions from industry, from cities, and from what you may call our modern way of life. So what has been done so far to tackle these problems, at least from a technical point of view? Well, many of the initiatives to tackle these problems are what you might call reactive approaches, okay? So you have a problem, and then many laboratories, and many of them incorporate into their agenda synthetic biology approaches, will try to reduce CO2 emissions. Different industries are trying to decarbonate their energy, go into electricity, for instance. Then instead of petroleum, there's this interest in developing biofuels. Instead of plastics, the idea is to have biodegradable polymers. Instead of chemical transformations, to go into biotransformations. So at the end, you have a sustainable production of various goods. You can improve crops in various ways. You can recycle. Okay, but if you think about it, all these things are what you may call reactive approaches. So that means we have a problem, the problem is there, then we try to do something about the problem. But can we take a different perspective and be proactive? So instead of reacting, we are proactive. Okay, then in that case, the challenge goes into what might be called design across scales. Okay, so the idea is how to go from solving a problem at a very small scale all the way to tackle a problem that might affect the whole planet. Okay, so I took this from a lady called Megan Palmer. So to put some frame into this discussion, one thing that is important is to understand how ecosystems respond to environmental stress, to environmental insults. So in many ecosystems, what we have is a linear or non-linear response to stress, but the shape of the response is such that when you stop stressing the system recovers the initial state. So you have a regime that is the sustainable functioning zone. You stress it by contamination, by CO2, by temperature, and then it might be stress and depart from this sustainable functioning zone. But when you stop the stress, it goes back. Good enough. Or you may have this non-linear response, but still, as you see, the shape of the curve is such that when the stress go back, then the regime B that is not very good goes back to regime A. The problem is, in many cases, when you have a situation where you have what is called a threshold-dependent catastrophe. In other words, that you are in a sustainable regime, you stress the system, and then the system, if it is too stressed, it goes beyond a tipping point, and then it goes into a situation under which stopping or decreasing the stress will not help to recover the earliest status. And in many cases, we have a problem with these tipping points. So this idea of being reactive apply better to these situations. If you have a lot of CO2, we can have less CO2. If you have a lot of plastics, we'll have less plastics. If you have a lot of problems, then let's stop the problems or let's mitigate the problems and things will be back again. But unfortunately, many of the situations associated to the current climatic crisis involve these threshold-dependent catastrophes. And scientists for a few years now have been able to identify a large number of locations where ecosystems have really passed through the tipping point to the point of making now the landscape not amenable to returning to normalcy just by stopping the cause of stress. And this is well documented. and I don't need to give you more details about that. So, but there are large parts of the planet that are already affected by many, say, tipping point trespassing, including, by the way, some problems in your country. Okay, so how can we think big and develop some technologies and concepts through which we can tackle and to some extent remediate or mitigate the problems associated to climate change, emissions, and this type of challenges? So one idea that has been put forward in the last few years is this concept of Earth terraforming. What is terraforming? Well, terraforming is this idea that you may have seen even in science fiction movies or science fiction books, in which you go to a planet with no life, and then you bring into the planet all the basic elements that are necessary for life, with the idea that you make a kind of seed cocktail of life components that will prosper and eventually will propagate to the point of create some life in a planet that otherwise would be dead. So the idea of these people that pioneered the concept is that, well, we don't have to go to Mars, or we don't have to go to another planet, because we already have on Earth locations that are so deteriorated that life might be impossible. So why don't we bring to Earth many of the concepts, many of the tools, many of the models that astrobiologists have developed for other planets? We bring them to Earth, and then from there we can perhaps propose solutions to similar problems that are in our very planet Earth, rather than going to another planet. So how can you apply this specifically to some of these tipping point problems that I just mentioned? Well, the idea here is that you have an area, you may have an area that is completely dead or semi-dead from an environmental point of view, and you have an area where vegetation, that is the basic, say, origin of everything else, can prosper. So there is a kind of borderline here between vegetation and desertification, and the idea is that you can inoculate, you can bring into the scenario some mixtures of microorganisms that by themselves or through some genetic modification can restart a process of life in this location that otherwise would not be possible to support life as it is, okay? So in this case, the idea, and this is how this concept crosses with synthetic biology, will be the following. What if we first identify the target of our action, then we just make a block diagram of what type of biological activities would be necessary for recovering life and developing a sustainable ecosystem in a place where the situation is not sustainable. Then we convert these block diagrams that are basically activities in, we look around what biological systems could be able to deliver these activities that we have previously planned, and we have planned their interactions as well. And then we can make an abstraction on how the interactions can be represented in terms of logic gates. And then finally, these logic gates can be implemented as genetic circuits that we develop and write in the DNA in plasmids or in the chromosomes of the biological agents at stake. Okay, so this will be a kind of vision that if you want to tackle this problem, then you first identify what activities you need, then you identify or try to look for biological platforms that could deliver these activities and also will deliver them following a given interaction blueprint. And then these biological systems have DNA, and the DNA can be reformatted and shaped in terms of genetic circuits, and then the genetic circuits can be just written in DNA. Okay, so at the end, the idea would be to connect synthetic biology here with environmental problems at large scale there. So what problems are out there that could be tackled through synthetic biology methods, and many of the methods that we have seen these days? Well, there's a list here of different problems, and let me say something about each of them, because each of them is very important. The first and most important is the issue of CO2 and greenhouse gases. And this is something that we see in the papers and in the press every day, I don't need to convince you that emissions of CO2 are very problematic because they have changed the climate at a planetary scale, and they are to some extent, or to a large extent, to be blamed for the increase in temperatures. But there are many other issues. For instance, we have the problem of arid ecosystems, drylands, and the challenge for synthetic biology will be to increase their humidity. We have the problem of plastic waste. We have the problem of micropollutants, in particular pharmaceuticals that go into the environment, and they are in very low concentrations, but still they have a big impact on trophic change. Then we have the problem of nitrogen. So most nitrogen that is used in agriculture has a chemical origin that the production of which is very impactful for the environment. So it would be good to have new ways of biological fixing of nitrogen. We have a problem with air. I mean, you just go to the main street there and immediately you notice that air pollution is quite an issue. There's a big problem with the management of lignocellulosic residues, the agricultural residues. We have many of them and we basically throw them out or burn them. So we have the problem of phosphorus. So phosphorus is an essential element of agriculture, and in a soluble form, it is obtainable only in a few locations in the whole world. When we run out of soluble phosphorus, there will be a big crisis in agriculture. Can we do something about it? We have the problem of water treatment. And finally here, we have the problem of scorched soil after fires. So you know that every summer in Australia, in the Mediterranean area, and also in California, you have these massive fires that burn out of wood. And it's not only that they produce a lot of CO2, it is only that the soil there is quite destroyed. Again, can synthetic biology do something about it? Okay, the answer to that, or the optimistic answer to that, is yes, but then we have to solve some technical problems and technical issues like follows. So first of all, we have to think on what functions could be necessary to restore ecosystems to the levels that they were before. And we can do that in the shape of a block diagram, but also we have to look at the components and program the components to be able to establish these interactions, okay? Then, once you have the macros that interact with a given blueprint, then you have to put them somewhere, and that somewhere, in many cases, is soil. And in soil, you have various matrices. You have soil, you have plant roots, you have soil stuff, you have humidity, you have other bacteria. So in that case, the challenge for synthetic biology is the design of surface interactions, microbe surface interactions. And on top of that, in many cases, bacteria are unable to do their job in isolation, and they have to collaborate with other partners. And therefore, the third challenge is the design of microbial consortia. If you remember, in previous classes of this course, we have dealt with technologies to bring this about, to technologies to bring this about, and with technologies to bring this about. So in principle, synthetic biology may open an avenue to really tackle these massive planetary problems with a new perspective. To do that, you need a chassis. And again, the term chassis has been used in this course various times. And depending on the ecosystem that you want to target, then you have different types of chassis here. So you have a chassis that might go for plant roots, other chassis, for instance, some cyanobacteria that will do better in drylands. Then you have other chassis that will do better in marine ecosystems. And you have other chassis like Pseudomonas putida that might do better in soil ecosystems, for instance, soil contaminated with plastics, okay? And in every case, you have to think on what activities, what artificial, what synthetic activities, what circuits you want to enter in each of these chassis to have them working exactly in the direction that you want them to use them. Well, you may say, well, this is very nice, but at the end you have a culture, or you have a petri dish, and the problem is how you spread these activities at a very large scale. Okay, so because as I said at the beginning, one thing is to observe and to produce a nice activity in a petri dish in a small bioreactor, and a completely different problem is to have everything or the activities of interest spread at a large level. So how can we do that? Well, to meet this problem or that challenge, this term has been coined environmental galenics, that is about formulation and spreading of symbio agents. I have to say something about the term galenics. So galenics in pharmacological science and in pharmacy science is the, say, science or is the technology to deliver specific medicaments to a sick body, okay? So it's not enough that you have an active molecule, that you have an antibiotic, for instance, or an immunomodulator or something. You need to deliver that through a vehicle, through a specific technology to put the activity in a given target. So you may have the same drug, and the same drug can be delivered through a pill, through an injection, through a suppository, through an aerosol, and so on. So this is the galenics that go into the health sector. Then environmental galenics is therefore the area that meets the challenge of producing biological agents in a form that can be delivered to a target ecosystem. So what type of forms these agents can take for delivery into a location? Well, you have different possibilities. You may have just monoclonal agents, a specific bacterium that has everything, all the circuits, everything just by itself. You can have consortia, and where you have engineers and type of interactions between them, or some division of labor, or some metabolic traits that have to be exposed or produced by the combination of various strains here. And you can have them forming also three-dimensional structures. Other possibility that we'll discuss in a minute is to just have DNA as the agent for delivery of activities of interest. And then in some cases, what you can do is to combine some of these activities with other carriers. And for instance, one possibility would be to develop bioremediation units where you have a component that might be a plant or even an insect or something that moves. Then you have a bacterium, you may have other bacteria, you may have other fungi. I mean, you can make this kind of consortia that, for instance, can be in some cases engineered to do something else apart of bringing some bioremediation activity, for instance, to counteract the effect of some pathogens in the plant. So at the end, you may have, I don't know, this bacterium degrading a given compound, this other bacterium providing some nutrients that are necessary for the bioremediation, and at the end, the plant providing some root exudates that will feed the whole system. And then at the same time, this bacterium could be programmed to kill pathogenic fungi. So this is the kind of, say, consortia that one can think of for having bioremediation units or biological, say, materials that can be sent to an ecosystem and then do better. Obviously, you need not just to have the bacteria put together, you need to have some modeling on how the different bacteria may behave when put together, what type of interactions they may have, and then what would be the reaction or the behavior of the system when you put them in a natural environment. And this is part of the agenda of modelers and more systems biologists, because you may have a pre-existing community there where you introduce a synthetic agent, and then you have to identify some parameters that ensure that when you put these new synthetic agents into the environment, you get into a situation here that is stable. So the bacterium does not displace all the community, nor the community displaces all the bacteria here, but you have a kind of nice, say, equilibrium, and the introduction of a new member of the community is stable rather than going in one direction or the other, okay? So this is another possibility, another branch of this endeavor of thinking big and putting synthetic biology agents into the environment. In what format you can put this type of things? Well, there are many propositions. One of them that we have ourselves play with is the idea of producing bacteria in flasks or bioreactors, but then freeze-drying the bacteria in capsules, in gelatin capsules. So gelatin capsules have been used in pharmacy for a long time, and the advantage of gelatin capsules is that they dissolve in water. So what we and others have done have been to produce bacteria in the shape of a powder, because they can be freeze-dried, and then the powder is in a machine encapsulated in this form, and then in this form can be sent to soil, for instance. The advantage in this case is that the shape of the of the capsule is similar to the shape of a seed, and therefore you can use agricultural technologies for spreading seeds to the same end of spreading capsules containing bioremediation agents. Furthermore, it is possible to enter in the capsule some formulation for increasing the effectiveness and for increases, in some cases, the containment of a particular agent in a given target. Because, for instance, you have a bacterium here that does something interesting, and you don't want the bacteria to escape much beyond the place where you inoculated, you can make this strain, for instance, axotrophic for a given amino acid or for a given metabolite in such a way that once the metabolite is consumed, the bacteria cannot spread much further. So these are tricks that have to do with the formulation of bacteria that are destined for the environment. This is just a little experiment. These bacteria have been programmed to degrade xylene, and they have been also engineered with a biosensor that will indicate that xylene is being degraded at the same time that they emit light, okay? So what this indicates is that by having a formulation of this type of bacteria, you put it in a capsule and you put it in soil, and then you see that over time you have a signal here indicating that toluene is being degraded. And in cases where you don't have toluene, well, there's nothing because there's no activity, okay? So this would be one of the cases where you apply these galenics that I told you before for the sake of putting interesting things into the environment and having them working. But you can think big, and instead of just thinking on capsules, you can think in other vehicles for large-scale spreading of activities, okay? So you can produce the bacterium of interest or the community thereof in a fermenter, but then when you have the cells, you can spread the cells with a plane, with an aerosol, or you can go through other channels. For instance, you can combine these bacteria with seeds, and bacteria in many cases colonize the surface of the seeds, and then you can use a tractor or a agricultural method to spread these things at a large scale. But you can think in other ways as well. So, for instance, you can engineer the microbiome of some insects to carry the bacteria of interest, and then these bacteria that colonize the insects will be able to spread the activity in different types of ecosystems. In some instances, it has been known, for instance, in the case of some larvae, that some larvae are able to chew up plastics. And therefore, if you engineer the microbiome of these bacteria, of these larvae, together with the microbiome in such a way that the microbiome contains plastic degrading bacteria, and then at the same time you have the mechanical effect of the insect, then at the end you have a kind of walking bioreactor that can do a much better job. But you can think also in other vehicles. You can think on clouds, for instance. Bacteria inhabit clouds, and in many cases bacteria are packed with clouds. Bacteria are packed in clouds. And for instance, there are many clouds that are very rich in methanotrophic bacteria, so they can eat methane. And there's a whole branch of technology that is called cloud engineering, not cloud in the sense of computers, real clouds. Real clouds are there, through which you can inoculate a cloud with a bacterium, and then because of the atmospheric movement of the cloud, then bacteria will be dispersed at various locations. Or you can even exploit these dust storms that in many cases can go through large areas and are sometimes coated by a large number of bacteria. So just 1 year ago or so, there was a dust storm in the Sahara Desert here that propagated all the way to the UK. And experiments and data indicated that through all the way, they were leaving behind many microorganisms. So as this phenomena will happen, will like it or not, then why not exploiting these currents, these natural channels of propagation of bacteria for our own end, for our own sake, right? But you can still give this one more screw turn. And then instead of thinking on propagating bacteria, why not thinking on propagating genes themselves that encode beneficial traits? So in this case, the idea would not be to have a bacterium of interest or a synthetic biology agent of interest and make it spread. In this case, the idea would be to propagate the DNA that encodes the trace of interest. So how can you do that? And what you want to spread? Well, there are some activities that will be super interesting to spread at a very large scale. So for instance, to counteract the effect of CO2 emissions, the key enzyme to do something about it is the enzyme called Rubisco. So Rubisco is one enzyme that is the principal enzyme in the photosynthetic, say, pathway. And what it makes is that it's able to take a pentose and stick a CO2 into a pentose. And then you create two intermediates here, and the CO2 is fixed. This is a critical reaction. And the critical reaction happens not only in plants, cyanobacteria, and other types of vegetables, it also happens in some autotrophic bacteria. In such a way that if we had a way to increase the general capacity of the biosphere to have more Rubisco activity, chances are that the whole biosphere will capture more CO2. The advantage of using this concept of propagation of DNA is that, as we know very well, bacteria and DNA propagate very quickly. So you may have something at the level of a genome, then it impacts the cell, it can propagate to a population, to a community, to a biofilm, to a niche, to a landscape. And we know that mostly because of the studies on the propagation of antibiotic resistance genes. So you may have a mutation in a genome, for instance, that originates a new gene for spreading or for antibiotic resistance, and then the gene appears in year 1, and 10 years later is all over the planet, okay? So that means that DNA propagates very, very quickly, and some trait that is invented at the level of a genome, given the right selection conditions, can propagate all the way to colonize an entire landscape and even to colonize the whole world. So in that respect, there's this interesting concept that is called fortification of environmental microbiomes with beneficial traits through massive horizontal gene transfer. So that means that you have a pre-existing community in the environment, and then instead of entering one more member in the community, what you do is that you spike the system with some plasmid donors or some super donors of DNA, and then they may not eventually survive, but the information that is encoded in the plasmids will propagate through the community, and eventually all members or many members of the community will be able to have that activity being expressed in their kind of activity portfolio. How can you do that in practical terms? Well, this is one project that we have in the laboratory with these people, and the idea is to see whether you can engineer one bacterium, inoculate that bacterium in an ecosystem, and then see whether at the end the trait that is engineered in a mobile element in one of the bacteria will propagate to other members of the community. Well, this process of horizontal gene transfer happens all the time in nature, and there are some plasmids that are naturally very promiscuous. Promiscuous means that they can self-transmit to other nearby bacteria, and they can also replicate because they have broad host range origins of replication, broad host range promoters, and they can move around in a community. They can circulate genes in a given community. So this plasmid is called RP4, is one of the most promiscuous plasmids known. And what we did was to make a small version of this plasmid and then make a mini plasmid that now we tested for propagation in a soil community, for instance. So what we made was a donor strain carrying this super promiscuous plasmid, inoculated in soil in a plant pot, and then we observed the transfer of this plasmid to different members of the community. It was easy because one of the recombinant genes that we engineered in the plasmid was GFP. So immediately you can detect transmission of the plasmid because you can really see fluorescent colonies appearing on a plate, okay? And what we observed is that the same plasmid could really go to many different types of recipients. Furthermore, this type of experiments reveal also that the donor itself over time disappear, but the number of bacteria that had acquired the plasmid increased over time, indicating that the concept was correct in the sense that we have a donor. The donor, we don't care about the fate. What we are interested is that it delivers its genes to the neighbors. And this is exactly what we see here, okay? So basically, this type of experiments indicated that not all members of the community receive the plasmid, but many members receive the plasmid of interest carrying recombinant genes, in this case GBFP, but in practical terms could be any other bacterium, and then the donor eventually disappears, okay? So this is a kind of concept that we want to nurture for future applications. You may also think, well, this is a bit crazy. How are we going to release things into the environment? But what happens in this case is that we can apply some criteria that has been developed earlier for clinical and health-related issues that are so-called clinical trial phases, phase 1, phase 2, phase 3, phase 4. So in medicine, when you have a new drug, then you apply first phase 1 to a small number of patients, then you go to phase 2. That means you increase the variety of patients that you deal with. Then phase 3, that is the most critical, you apply this medicament to the actual patients that might be helped by that treatment. And then on that basis and the outcome, then the medicament is approved or not. And then you can make models to verify and to monitor the outcome of the different applications or the different treatments. So in environmental, say, releases or environmental applications, we can do something similar. We can start with tests at the level of Petri dishes. We can go to something called microcosms. Then we can go to another level that is called ecotrons, where you have in a small ecosystem with everything that you need. You have plants, you have water, you have soil, but everything is encapsulated to control all the parameters, and you can monitor what is going on inside here. And then depending on the outcome of this phase, then you can decide to go big and to implement the treatment at a large scale or not, okay? So this is something that is currently developing, because to this day, this type of large-scale environmental releases and large-scale environmental application has not been made yet. Well, you can say, well, this is very nice, but maybe not everything is for a good purpose. So and maybe there are other possibilities to use synthetic biology and synthetic biology for at a large scale. And well, reality is that this is a technology, as is the case in any technology. Maybe the technology is not itself neutral, but depending on the application, it can be better or worse. So different countries have their own programs on application of synthetic biology to defense or to war. Okay, so to war purposes. And this is a slide that I got from the corresponding agency in the UK, where you see that different synthetic materials and different synthetically produced organisms can be used for various, say, defense applications. So this is one thing. But at the same time, it is also possible that synthetic biology, because of these possibilities to intervene ecosystems at a large scale, then can be weaponized for peace. And recent studies have made lists of what type of problems that used to be associated to political crisis and to war or peace situations, what problems could be really tackled through synthetic biology-based microbial solutions. You have hunger, you have productivity, you have environmental pollution, you have clean water, you have poverty, blah, blah, blah. And then you can make a list of possibilities of solution, or at least approaches that could tackle problems that otherwise would be very difficult to handle. Okay, so that opens a completely new possibility, because, so far, before the times of synthetic biology, then all we could deal with was with the extant biological world. The extant biological world is the one that we see around us, and that, is what it is. I mean, you have a tree and use the tree as it is. You have a product, you have to use that product. But the kind of challenge, the kind of perspective of synthetic biology is that it opens the possibility of liberating biology of its own limitations. And this is something that you may have get the intuition in some of the lessons before. If you remember, we had we had a lesson or a lecture on the standard genetic code. And then, well, the standard genetic code is prefixed by evolution, period. So before synthetic biology, we had the 60-some codons, and then each of them, or various of them, codified by proline or by histone or whatever, and it was given, and there's nothing we could do about it. However, we can kind of emancipate biological systems from its own limitations, and then think on life forms that have a much wider scope of possibilities and applications that you have in the natural world. Okay, so this is one important aspect. And therefore, that takes us to another, if you want, I wouldn't say more philosophical, but more conceptual issue, and is how can we design, can we use biology, I mean, can biology be designed, and also can we use design to decide what we should or why should not do. So this starts, this kind of idea or this kind of question take us to raise the interface between synthetic biology and art. Okay, so art is not necessarily what we consider always art, like paintings or like buildings. In many cases, art is about exploration of reality, and also how this exploration may propose other, say, scenarios for our life different of what we know. So from the very beginning in synthetic biology has been this sub-population or this sub-community interested in thinking and implementing, in some cases, the consequences of synthetic biology in the world of art, in the world of aesthetics, and also in the social, say, framework. So the key questions here are, can we design nature? How do you design nature? But how you can design nature well? Okay, so these are questions that may sound philosophical, but depending on your answer, you may have completely different scenarios. So one of the questions that are brought about for this type of artistic explorations is that we take for granted that in biology things are as they are, and that there's no way to have them working or structured or performing differently. So there's a bunch of different artists that say, well, given that now we can program biological systems in our own desire, so can we generate situations where the biological logic is different of the standard logic as we know it? Okay. Okay, well, there are various possibilities for that. So there's a whole branch of art that basically gets inspiration from biological systems, mostly by biological shapes. Okay, so this would be a typical biological, say, form. It comes from some cover of carbon deposited calcium carbonate in a bacterium or something like that, and or some exoskeleton of some insect. I mean, these are forms that are given by nature. And then some artists can say, well, maybe we can start with some forms as given by nature. We can digitalize the forms, and then on that basis, generate objects that are inspired in terms of the shapes by something that nature has given to us. But they are not necessarily what we have at the beginning is different, but we get inspiration of a form given by nature to create something a bit different. Okay, so this will be the result. There will be a 3D printing of a form that we find in nature, and then this will be the artistic abstraction or the artistic rendition of what the artist has seen here as the essence of this particular structure. But you can go much further, and in many cases, apply things that happen in the biological world to many other, say, dimensions. So in many cases, well, every project that we have as humans go like you have a starting point and you have an end point, and this is where you think your project will end up, starting point, original scientific hypothesis. But then in between, I mean, you have all types of issues coming in between, and this is the typical situation, for instance, for grad students, and later we will have some grad students telling us their stories. So you know where you start, but you think you are going to end here, but at the end you end in a place completely different. Okay, and in between, many unexpected things happen. Well, this is something that can be applied in different ways in the artistic endeavor and in the design endeavor. And for instance, in this case, what we have is a project in which someone tried to produce glasses or cups on the basis of a mycelia that we wrote here on a given geometrical form. Okay, so the question is, for how many materials we can use to have a cup, a water cup? Well, you say, well, we have glass, we have plastic, we have paper, but can we have also an organic product, an organic object that delivers the same activity, the same function of other materials, but at the same time is not made with those materials, but with something that is alive or has a life origin? Well, this will be an artistic exploration, right? So this is not a product that you can buy in the supermarket, but this is kind of an exploration of reality, exploration of the possibilities of synthetic biology with an artistic perspective. Same thing, if you remember, we discussed these optogenetic systems that in many cases enable you to convert bacteria into a photographic film, and we have seen that before. So can we do that? Obviously, there's a scientific motivation for that, but it is also a kind of artistic challenge, because in that case, artists can produce shapes with different materials. So artists never thought that they could use bacteria as instead of paintings or instead of photographic films for making shapes or for developing different structures. But there's something still more challenging, okay, that you might be kind of surprised, and it's the following. So, you know, in the extant biology, we are used to our bodies, and we are used to the cells, and in the cells and the body, you have an in and you have an out, okay? So, is that like that, or can we think differently? Can we think on a cell or on a body that is just the contrary, where what we have in is out and vice versa? I mean that, okay, so you say, well, no way. Well, wait a minute, let's give it a try. So one of the, I mean, one way of provoking this type of thoughts would be, for instance, to start with the microbiome. Okay, so the microbiome is in your body, and then you know that depending on the part that you have in your body, you have different bacterial communities and so on and so on and so on. Okay, so then, you know, on the other hand, you have cheese. Well, I know that in India, this type of cheese is not very common, but, you know, in Europe and in the US, people love cheese. And this cheese and different types of cheeses are made on the basis of microorganisms that are isolated from soil and that have been observed during millennia that produce at the end some nice edible products. But say, well, so this is the tradition, okay? So can we think on making cheese in a different way? Okay, so for instance, you know, are we limited to the bacteria in the soil or to the traditional bacteria for producing cheese? So an artistic team say, well, let's try to get cheese out of microbiomes that have not so far been explored as the source of cheese production, for instance, feet or different parts of the world. Okay, so what these people did, and Christina Agapakis is one of the most provocative ladies in this respect, is to make an experiment in which they just went through the bodies of various people, to the mouth, through the belly button, so the mouth, so the toes, and on that, the basis of the bacteria and the microbiomes that they were isolating from these locations, then they made some different cheese fermentations, okay? What do you think about it? Okay, in principle, it looks like very disgusting, right? So, however, they took this to a cheese factory, and it was incredibly successful, okay? So they were able to sell all these cheeses at a very high price and with consideration by the customers, like being a very good product, because they had gone beyond the limits of the standard ideas of what doing cheese is like, okay? So this idea of kind of going beyond the limits sometimes is unsettling, but at the end, it may pay. Okay, along the same lines, then what could we do for dealing with large environmental problems, well, we have said before that we have ecosystems that are deteriorated by various things and so. So how can we start with a deteriorated system, a deteriorated ecosystem, and then return to something that looks like a wonderful jungle, like a wonderful place with greens and a kind of original, say, scenario? Well, at a distance, you may think that this is very nice because it looks like very natural. But in fact, if you look closer, you see some little balls here, some little things here. And the reason why this is maintained in such a good shape is not because we just let nature to do the job, but because we help nature to do the job. And we do that because we have engineered in the laboratory. This is an artistic simulation, okay? So this is not something that happens right now. But in principle, synthetic biology could deliver or could produce, for instance, mobile bioremediation units where you have an animal or an animal-like form that is constructed, is artificial, and well, it's just a device for soil bioremediation, and it has all the nice features that you would like to have in a bioremediation agent. It will have like photosynthetic cells on the surface so that it does not depend for energy on having to eat strange things. So it's programmed towards acid soil. It's designed to have some features in the DNA not to be edible by other organisms in the system. So this is something that you can engineer, and with the tools of synthetic biology. This is something that has not been done yet, but there's no theoretical reason why you should not do it. You can engineer a worm, you can engineer a microbiome, you can engineer cells on the surface that capture light. I mean, you can really engineer a biological-like agent able to make some degree of bioremediation and helping to maintain the large ecosystem. Same thing, you can make, for instance, a self-inflating antipathogenic membrane pump that can go to the barks of the trees, and they may inject some compounds in the barks that will help say will help the health of the trees. And again, you have a description here. You can patent if you want, with all the features and everything, where you combine different parts, different devices, different circuits, and at the end, the compound or this agent or this self-inflating pump will be able to protect some of the plants from pathogens that may be caused for pollution. And again, you may have a blueprint, all the components before and after. So this is something that in synthetic biology could be happening and is at hand. It's not a dream. Okay. Similarly, you may have these other kind of strange shapes, and these strange shapes might be, for instance, an autonomous seed disperser. Again, you have all the devices, all the circuits, everything in such a way that this animal-looking agent that doesn't have to be an animal, it can be completely artificial, can go around and is endowed with all types of properties here to secure that some seeds are permanently being released into the woods, and therefore the homeostasis of the ecosystem is maintained. Similarly, for instance, you can engineer plant leaves so that you have a biofilm that captures pollutants, for instance, and then you may have plants in your living room or in your house or wherever that have been engineered to have a biofilm, or themselves, their biomass, have catalytic activities able to capture pollutants from the environment. I mean, you can really go on and on and on and on, and at the end, you may have a set of agents that can really make a big difference in terms of environmental preservation. Okay, so this is something that is, I insist that these are renditions. They are not real, say, things, but well, you can think big and start considering that things as they are doesn't have to be like that forever, and that you can be imaginative and rethink methods, products, interactions with the environment that you have that might be different. For instance, one of the projects that I find more interesting is this idea of reformulating maggots for therapy. You know that maggots are these worms that eat dead flesh, and the virtue of these bugs is that they distinguish between dead meat, dead skin or dead flesh versus live one, you know. So you have a wound that is infected, and then you have some part of the tissue that is dead and the other is alive. These worms will just eat the dead tissue and will leave behind the good tissue. I mean, again, this is something like in the case of the cheeses that, you know, doesn't look very nice, but it can be as effective as other approaches that require surgery or they require other things. Same with earthworms. So earthworms go into the soil. They can, in many cases, penetrate into the inner layers of the soil, and then this will be a perfect bioreactor because you know, these shapes have, I mean, these worms have a big mouth, and they eat the soil, and the soil can be passed then through their gut, and then if the gut is colonized by bacteria that have been programmed to produce good bioremediation activities, then you may have a super efficient way of remediating soil in many cases. Okay, so so the type of possibilities that synthetic biology offers for the future are just immense. Okay, so this is something remarkable. And well, in some cases you can make actual, say, experiments. In other cases, you can make simulations and predictions like this that you have here. In this case, for instance, you have this gentleman here that looks like Indian. I don't know where he comes from. And he's smelling a culture of E. coli that has been engineered to smell banana. Okay. So I don't know whether you work in the laboratory on a regular basis, but you know that for bacteria, I mean, to monitor bacterial growth, you have to monitor once in a while the so-called the turbidity. Okay, you have an apparatus, and then the more turbid the water is or the liquid is, you know that the bacteria are growing better. But then someone say, well, why don't we want to have this super expensive equipment? What we do is that we make a pathway for production of this banana smell that is associated to a type of alcohol for production of which we know exactly the pathway that I have to take, and then we put it in front of a promoter that is responsive to a stationary phase. So that means that instead of making measurements of your culture on the basis of the turbidity, what you do is that you smell your culture, and when the culture smells banana, you know that you are in stationary phase. Okay, so there's a kind of what is called frugal technology. So frugal technologies is this idea of replacing super complicated technologies by something that is equally effective, but they are not, that is not so complicated. Also, and this is reality, this strain has been made. This is a simulation, and the simulation is to use bacterial mycelia as construction materials. Okay, so imagine a situation where at some point you have this massive mycelia used for construction. This is something that is a simulation, but indeed there are companies in the US that produce mycelia as isolation products for construction. Or, for instance, in this case, you have this IM project that I mentioned one of the first day called E. chromai instead of E. coli, in which the E. coli strain that is eaten and introduced into your gut has been engineered to give you different colors depending on your health conditions. So if your feces are green, that means that you are okay. If it is red, maybe you have diabetes. If you have this, maybe you have high blood pressure. You have that, maybe you have a flu. I mean, this is the type of diagnostic, perspectives that are open again for synthetic biology. But this is not the end, and obviously there are other, aspects that propagate into the society. Don't think that synthetic biology is just all about biotechnology. It also questions many of the common ways of interacting with our natural medium. And for instance, there are some, again, simulations in which some artists or some people interested in anticipation and prospective future, for instance, think that you know, you can generate a kind of artificial island that could create would claim independence, political independence, because you have engineered a platform that can produce a biofuel, a diesel, okay? And in such a way that it could be autonomous, can float all around, and it will not be under the jurisdiction of any country, and it will, you know, self-sustain because of the high capacity to produce energy just by itself. You can produce machines. There are some, say, materials that can be produced with bacterial leather. I will return to that later. And there are many possibilities of engineering, say, new objects, new functionalities on the basis of materials that have a microbial and in many cases engineer origin that were not even considered at all to be possible in the past. Okay, so for instance, you have this. This is made of bacterial leather, these biofilms that are dried and treated. And in this case, you can have a cup or you may have a container for various uses. So that's something that can be done easily. Also, you know, artists are quite fascinated by the fact that you can program surfaces covered with bacteria programmed to respond to given inputs for generation of various forms and exploration of the limits and definition of boundaries. I mean, this is things that belong to the artistic program always, you know. So artists, in particular plastic artists, are always very interested to explore forms and how forms can be created and so. And the fact that you can use bacteria as elements for these forms, in many cases, fascinate them because they see forms that they have not seen before. Okay, so then in this case, the question that I leave you with is: can experimental design take a more active, critical, or even guiding a role in our habitary future? It's a question that is not it's a question, is not that answer, and probably your generation and future generation will have to deal with it. One, step in that direction is to recruit artists to laboratories. I don't know whether you have that programs here, but in Europe, it's becoming very popular to incorporate artists to different, laboratories for a while, for a month, for 2 months, or whatever, and then ask them, well, you be here, you talk to people, people tell you about your project, about the projects and so on. What type of artistic inspiration you get from this, okay? So in my laboratory, we had this lady that's called Lara Tabet, that is a graphic and plastic artist from Lebanon. And then she came to my laboratory for some time. And then after a while, she kind of proposed interesting projects. There's a video there that perhaps I would like to show you. Video, let me see.